Okay, thank you. Maybe I can start by giving a few introduction words for this meeting. So first of all, thank you so much for being here, all of you. Nice to see you for those who I already know, and welcome for the other ones. So as a recap, this meeting is part of our working group on subtask 2.2.1 of Biodiversa Plus on the harmonization of monitoring protocols. And here, this is the first meeting on a series of sessions that we're trying to plan of experience sharing. So the idea is really to provide the space and time, even virtually, to share your experience with a given protocol that you are implementing or that you want to discuss. And so the other participants can ask questions and see insights of possibility for them applying, or just trying to understand better the choices that are made with regarding the protocols for biodiversity monitoring. And so please feel free to ask any questions. This is safe. It will be recorded and available on the Biodiversa Plus YouTube channel. But the idea really is to have this space for exchanges between um, people working in biodiversity monitoring and implementing protocols. So here our first guest is Julia Sieber, who has agreed to present the protocol on the soil biodiversity monitoring, and we're really happy to have her here. She's one of our active partners in this working group, and she is really great with that and has brought a lot of insight as well. So thank you, Julia. The floor is yours. You can go ahead.
Thank you, Michelle. So I'm happy to present today the protocol for field sampling we are using in the soil biodiversity pilot study in the biodiversity network. So we are not yet monitoring, but on the way to pave the way to the monitoring. So I will present what is like a bit of, see if it goes on here, a bit of a general context, why we do this, then tell you a little bit about our protocol, and then the main challenge that comes with that protocol and what we saw after doing 1 year of soil biodiversity monitoring in the biodiversity network. So the general context, why set up a soil biodiversity monitoring at all? Beside the fact, of course, that we all rely on soil biodiversity, of course. It is one of the priorities of the Biodiversa partnership, and it was chosen as a pilot study in the first wave of pilots. So we are already in the second year here. And well, there are actually 2 initiatives already out there. There are quite a lot of national monitorings in Europe, but they are kind of 2 different things, so it's not really possible to compare them. And what we really need in soil biodiversity is a harmonized program and a harmonized protocol and a program across Europe. So there are 2 bigger initiatives already out there. One is the LUCAS. This is a European initiative done by the JRC, and it samples and analyzes the main properties of topsoil, and actually since 2018 it also does soil biodiversity. But even though it has really a lot of sites in Europe, it focuses on agriculturally used soils, so no like mountain soils, no really natural habitats. The second one that is out there is Soilborne, with its 2 add-on modules, Soilborne Food Web and Soilborne Earthworms. This is a global initiative, and it uses a paired approach, which means that it always chooses a pair of sites. One is inside a protected area and one is outside a protected area. But since it's a global initiative, there are only few sites in Europe. So even though they are big initiatives, they don't really give us information on soil biodiversity across Europe. So that's why we launched our pilot study to maybe do a soil biodiversity monitoring in Europe. And in terms of EBVs, what might come out of that pilot or out of that biodiversity monitoring, I had a look at the list of essential biodiversity variables that we might help to use. There's the one that is called functional or taxonomic diversity of soil biota, but I think in the latest version, actually, it only says now functional diversity. Okay. And I have to say that what we are doing is more the taxonomic diversity. So I'm not yet quite sure if in the end there's a specific EBV coming out of that monitoring and that pilot. But maybe to explain what will come out of our monitoring program, it's two things, because one of our aims is actually also to decide on specific methods in the end, and we are comparing the more traditional methods used in like pitfall traps and hand sorting of soil cores with the more modern, let's say, more molecular methods in terms of eDNA. So what the information that we will have in the end is from the traditional methods, it's abundances of selected taxa, but in that case, it will very rarely be on species level because of the really huge diversity of soil organisms and soil invertebrates. It's simply not possible in the monitoring program to go to species level morphologically. So we will have abundances, but more likely on order, or if it all goes well, on family level. But still, also a bit of community composition. From the eDNA methods, we will have a really huge diversity covered because we can do libraries on archaea, bacteria, fungi, invertebrates. But since the reference databases of eDNA are not really good at the moment, especially for soil biodiversity, we can more or less only tell about alpha diversity, but not community composition, not yet at the moment. So also in terms of data aggregation, all our data will be on site level because we are using composite samples in the pilot. The exception are the traditional methods, because then we have on pitfall trap level or soil core level, so really on the trap level. But mostly, the most of our data will be on site level. So what to do with all the data we are creating? Of course, we do reporting. We already have one report out there if you want to have a look, the first year report of the pilot study. But of course, it will be becoming even more important in the next years as the Biodiversity Strategy 2030 will come in place of the European Union, and this will also include the soil strategy. So in terms of soil biodiversity, it will be important for both soil strategy and biodiversity strategy. And last but not least, we have, I think, a well-designed protocol and sampling design, so it can also be used for scientific research. This is just an example of what we did with the data of the first year. And you can see that taxon richness differs quite a lot between different forest types. And this will be very important because altogether we still know way too little about soil biodiversity in general, and especially also in Europe and in the different habitats. So I think either the pilot itself or the possible monitoring that will come out of the pilot will be of high interest also to the scientific community. So now to the protocol. The two big initiatives, Lucas and Soilbon, use more or less the same protocol for fieldwork, and that's why we also use it. And I will explain it a bit more in detail, but the data that we are asking for from our partners in the pilot is, for one, to give us some background information on the sites they are surveying. So we ask them to give us the GPS coordinates, some information information on geology, the soil type, the forest type, and we ask them to do a very simple botanical survey. So this is data they give to us, but everything else is actually a physical sample they are sending to us at URAC, because we at URAC, we act as a, let's say, central hub for all the samples that are being taken in that pilot study. And what we are doing is we analyze the most important soil properties for each of the sites, which will be used as predictors in any analysis. And then, of course, the soil organisms. We do a pre-sorting here at URAC, so mostly family level, from the pitfall traps and from the hand sorting. And for selected taxa, we have an external company who do species identification for us. So maybe, for example, for spiders or for carabid beetles. And then we also have an external company doing the eDNA analysis, because we do not do that at our labs. So we really send the soil sample to that company, and they then give us back the results from the eDNA libraries, and we do 4 of them. So we really cover all phyla of soil organisms, starting from the microbes to all the invertebrates that are out there. So that all our partners know what to do. For that, we prepared a step-by-step protocol. And that was actually already the first challenge we had, because we were thinking we were quite detailed in that protocol, but in fact, obviously we were not, because there were then still quite some questions about it. And then we had a workshop at the end of the first year, and this is already what part of the version 2 of that step protocol, where we included much more detail for every step.
Sure.
So our partners can just take that one file, that protocol, where we really, in hopefully very much detail, explain every single step on what to do in the field. So that starts off with the sampling, choosing the sampling area, and then do the botanical survey, put the pitfall traps, hand sort the soil cores, and you could just click on what you have already done. And if you really want to have more detail, we refer to the respective pages in the Soilborne and the Lucas protocol. Additionally, we prepared a list of material, because that was very important, so that people know what to buy in advance and what to take in the field, because once you are in the field and something is missing, it's quite difficult, because most likely you cannot do your field work and usually have to go quite far. So we tried really to be very detailed also in that list of material that everyone needed for each specific step in the protocol. So just to give short glimpse on how that looked like, this is the first step, doing the botanical survey. So everyone going out and picking a central spot in that forest you want to sample, and then putting a square with 30 times 30 meters around that central plot. And of course, due to topography or heterogeneity, it's not always possible to do a perfect 30 times 30 square. So we now also give detail on how to deal with such difficulties if the topography tells you, well, it's probably not the 30 times 30 square. From that central spot, we ask everyone to do photographs of in each cardinal direction, so that we at URAC get an idea on how these forests look like, and so we get more an idea also on the topography of the forest without having to go there. And then we ask just for a list of plants in that 30 times 30 square. And I will not go into detail on every step, but the next thing they have to take 9 small cores, which are homogenized in one bag, so that we have a composite sample of soil from each of the sites, and that is then sent to us. And from that composite sample, we do the eDNA analysis and also the soil property analysis. Then we ask them to install 3 pitfall traps. We tell them how to do this, and they have to be left in the field for 2 weeks. After 2 weeks, they have to come back and get them again, and also how to hand sort soil cores of a specific size. So once the partners have done all that work in the field, they send all these samples to us at URAC, and we then process the samples further on. So the pre-sorting, the sending to the external company, everything. So we decided in that pilot that it makes sense to have a central hub doing everything else, because then it's done in a also harmonized way, because you never know that there's a lot of difference in labs doing certain analysis, and it was very important that the data is comparable. Yeah, well, the biggest challenge probably from year 1 was, as discussed before, the details of the protocol, so that everyone really knows what to do. But we also had a lot of administrative legal issues to solve, because maybe that's a specific thing of the soil pilot, because we have to deal with physical samples, and physical samples have to be sent around. So you all probably know about the Nagoya Protocol. Some countries have signed the protocol, some have not. So every partner had to contact the national focal point of the Nagoya Protocol to understand if anything has to be done about it, or at least to have a written, like an email or a letter stating that nothing has to be done or everything had been done. And also we had to come up with a material transfer agreement, which allowed all the partners to send the material to us, where we specified at each step of the analysis that we are going to do. That is very specific, because on the one hand, we do a genetic analysis with the eDNA, but since it's also only a taxonomic analysis, it's actually not that difficult. But that step, doing that contacting of the national focal points and signing the material transfer agreement, already took us quite a while. Since we asked to sample protected areas, many of our partners reported back that obtaining sampling permits is not always easy. And because we also have partners from outside of Europe, but it's also inside of Europe, it's really sending and receiving such physical samples, and especially concerning soil, is regulated in very different ways in every country. So because it could contain pathogens, or because it's alcohol and invertebrates, it was not always easy to really receive the samples, or also for us to send samples. So all that part was, in the first year of the pilot, quite probably the most difficult thing to solve. Yeah, that was what I wanted to tell you about our protocol, and I'm happy to take any questions.
Thank you so much, Julia. That was very interesting to have this more in detail view of the protocol, better than just a report, which we will all read, I hope.
Yeah. You don't have to, but you can.
I have a few questions for you. If no one else has one first, I might just go ahead, and then we can open the floor for any other questions. So my first one was concerning one of the main challenges that you mentioned, of the difficulty of writing the step-by-step protocol. And I wanted to ask how long it was as a process, and if it was a collaborative process with all the partners involved in the pilot, or how was this carried out?
Well, I have to say that the first wave of pilots, they didn't really have a lot of time to prepare the pilots, so it we just we talked to the Soilborn people, and they even did a training for us and our partners. So we more or less took the Soilborn protocol and tried to summarize the important information that is written in there in that one file, step-by-step protocol. And then we gave that to our partners. But then immediately, questions came in on how do I do that and how do I do that, and we answered all that by email. So that took probably 3 or 4 months until everyone more or less knew what to do. But when we met then in autumn for the final workshop of the first year, there was still a lot of open questions. So we kind of collected them and then wrote that second version of the protocol, trying to really include all the details that would be necessary. And now that version 2 is being used now in the field, so I haven't heard, or mostly haven't heard of any partners, maybe 1 or 2 questions still. So I'm hoping that we are there in the level of detail that is needed. But if I could put everything together, it was a process of a year.
Okay, yeah, it's big work to carry out something like this. That's great. And yeah, so you mentioned that you basically took the soil bond protocol and also with Lucas soil. And what did you adapt mostly or didn't adapt or for this pilot?
So the protocol of Lucas and Soilborn are more or less exactly the same. The only thing that they differ in is the depth of the soil to be taken, and there we decided to do it as Soilborn says. For the field protocol, we didn't change anything. We just added details on how to explain how to do something. But the protocol, as we use it now, is still the same as the Soilborn protocol as it was originally.
Okay, great. And did you have to readapt the protocol apart from details and the step-by-step protocol, but to change some parts of the protocol from the first year to the second year?
Only that because we do a repetition of the same program, we don't have to repeat everything. So like step 1 is the botanical survey, we don't have to do it twice, and also taking the composite sample for soil properties, we don't repeat that. So it's still the protocol states to do it, but we told everyone you don't have to do step 1 and step 2 because it's not necessary.
Okay, great. And I see there is Ulla who has a question. Please go ahead.
Well, perhaps, partly a question, but also a general comment. I think one of the problems when you discuss this kind of standardization is if you envision this soil monitoring biodiversity monitoring program to be a program apart from other monitoring activities in high nature value areas, or if it should be seen as a module, because that may put limits for logistic reasons and economic reasons on what you prioritize standardization about. For example, if you have a more general sample-based program of the Lucas type, if you have very stringent demands on which period of year you are taking the soil samples or putting out the traps, it make the costs rising, and it may be conflicts to other kind of investigations that are better done in other seasons of the year. So that may be a problem if you are going to optimize monitoring on the higher level in the future. Another thing I thought about that, but that may be a mistake, a specific case. You showed, I should have read that because we are part now on the second year, but I haven't read the details in the manual. But you showed this soil cores, coring to take the soil samples, but in other situations, I think it's not possible to use that tool. You have to dig by a shovel instead because it's too much stones in the ground. That is, I think, the situation in most forests in Sweden because we have a forest soil dominated by stony till soil types. But you perhaps, I think that Lucas have the option to have different methods in different situations there, and perhaps this was a specific case you showed where you where you can use this soil coring, what do you call it, boring down.
Yeah, I think it's a soil core. Yeah.
Yeah, but that is not possible if it is too stony in the ground. Yeah.
Well, yeah, both of your points are, of course, good points. The timing of sampling is, of course, very important, especially for soil biodiversity, because that changes throughout the season. That's why for the pilot study, we said to do 2 seasons. That would be like spring and autumn. But of course, when is spring and when is autumn in the different countries? So we kind of defined that as the peak vegetation of the ground layer in the forests, but that is kind of that gives you a certain time period when to do the sampling, but not that could change between years, and it depends on how the winter was and how the spring develops. So this is a very difficult point to decide on how often and when to do the sampling. So that is one thing that for a really national program would have to be decided how to deal with that, with the huge seasonal variability of soil organisms. And then the other point is also a very important point, and the picture I showed in here, if I can go back, this one is of course a very good example on where it is easily done to take such a soil sample, but we also here in the region of Bolzano have completely different forests where you barely get into the soil, and because it's so rocky. But that is a general thing of soil, and I think we need to be a bit open-minded about how to take the samples and how to deal with topography and certain characteristics of sites. And yes, I think the local protocol already gives some alternatives on how to take the samples then. We in the pilot decided if it's not possible to go as deep as we said we would, to obtain really the minimum amount of soil we need, then to take 2 samples next to each other instead of going into the depth. But that was already also a discussion point.
Maybe to follow up a question on that, from what I understand, this protocol allows quite some flexibility on the site selection. So it's each partner who decides where to do the sample, following some criteria, of course. How has this adaptability worked for the partners and for you? Is it easier this way as a choice?
Well, it's for the pilot specifically, we had bilateral meetings with each partner, and we had a wish list of forest types that we would like them to sample. But of course, the final selection was up to the partners, because the countries, like for example Germany, they are huge, and if they have to select 5 sites within that huge country, it's a bit up to them what is feasible. With all the driving, and for the pitfall traps, you have to go there twice, and then you have to do it in spring and in autumn. Of course, that needs some flexibility, and I think with all the partners, we more or less found a good compromise. not sure how that would really work in a transnational monitoring, because there you would probably have to decide on very specific habitats, or Lucas does it completely differently. It puts a grid on Europe, and within each grid there has to be a certain point which is sampled. So I guess there are different approaches, and I don't really have a good opinion at the moment on what is best.
Okay, thank you. Gabi, go ahead.
Yes, thank you, Julia. That was really interesting. I wondered about the administrative problems you told about. Is it the same for Lucas and Soilborn? Do they also use a central lab, and did they face the same problems? Because I think, especially for those joint undertakings, it's really a shame that everyone has these problems. And yeah, it would be great if, I don't know, initiative would be pushed to the European Commission that it, yeah, should be easier for, especially for joint projects. Can you comment on this?
Yes. So from the legal side, that was one of our recommendations, that actually we would wish that the European Union would kind of give us the legal perspective on how to deal with that. I know that both Soilborn and Lucas have a central facility which collects all the samples. Soilborn has then partner labs which do one certain analysis on all the samples so that you can guarantee comparability. That's why also we decided to be the central hub and then to do all the analysis in one lab. Also, Lucas does it like this. For example, the eDNA is done in, I think, in Lithuania. I think there's a big lab doing all the eDNA work. And all that legal advice actually comes from Soilborn, because when we started the pilot, I wasn't even aware that I have to deal with all that administrative and legal issues. So they were really a big help, and we also got the MTA, the Material Transfer Agreement, from them. We just had to adapt it to our pilot study. But having someone already knowing a lot about the different regulations in Europe was a big help. But if such a monitoring should really come in place in Europe, then it has to be more simple. It cannot be like as it is in the pilot. It would be way too much effort on administrative levels.
Ola, do you want to react on that maybe? I see you have your hand raised.
Yes, I want. I think this kind of centralization of the chemical, or by, in this case, more, for example, eDNA or biological analysis of the samples, and is a really a thing where you have often can have quite a lot of advantages of centralizing, because you can save resources on the national level. On the other hand, I think it's often the case, I guess it's the same in this case, that you collect often an excess of samples compared to what you use in the primary analysis you want to do in the samples. And then the next question will be, who will own the excess of the samples? And because you may want to do at the national level some other analysis at a later point, and then it's important to regulate that the country that have made the sampling, especially if it's mostly funded by the country itself in the future program. They should have the right to have the first say about who to use the remaining parts of the sample, as long as it's sufficient, so to say. So there's quite a lot of legal questions to delve into, I think, to avoid future conflicts. But it's on the other hand, as I said, it's much to be gained probably in some cases by centralizing some of this analyze work. For example, avoiding to have a much intercomparison between different lab with different analysis routines, etc. I think that is maybe a problem in DNA. Also, there's different extraction routines, for example, of the DNA. So there's really a balance, I think, that should be investigated in the future.
Yes, I completely agree with that. And if you look more into the traditional methods, it might be easier to do that on a national level anyway, because the taxonomic expertise is more in the country itself and not in a centralized facility. Like we at URAC, I don't know anything about carabid beetles of Turkey or the Azores. So if you really look at species, it might be more make more sense to do that on a national basis and then just collect the data. But what concerning molecular methods and also any soil property analysis, it is just known that there are differences between labs, even though they might use the exact same protocol. And to avoid having a bit of a bias in your data due to different labs, I think the solution for that is only that you have to use a centralized lab. But then, of course, that it needs to be solved who owns those samples and who owns the data. And also, I know that JRC has a very big facility storing the samples so that they can be reused for any other analysis. But JRC probably has the means to, like also the storage means, but we at URAC, we would not. I cannot store thousands of samples so that if like Germany comes to me and says they want some of the samples for any reanalysis so that I can give them back. That is for something like a soil pilot, really using physical samples, a big issue.
Go ahead, Ron.
Yes, thank you. It's quite an interesting discussion on aspects you don't often think about when you're not doing this kind of work. But you were talking about introducing a bias when you have different labs, but shipping itself already also will introduce a bias, the treatment of the sample. So there's so many aspects that can influence the result that you get in the end, that it's unclear to me whether it would be more worthwhile to do some more capacity building in a few different countries. So you have less shipping and that part is easier, as Nola said also, then they keep their own samples. Another thing I never thought about. So I'm curious in the end if you'll manage to get a better idea of where the biases are and what would be overall for a transnational monitoring program the best strategy, because it sounds easy to send something to one single institute. What you're showing that it's not that simple. In fact, it's quite difficult. It's very difficult. So maybe from the European level, they should push it a little bit more towards capacity building, making sure that at least everybody knows how to treat the samples, take into account there will be a little bit of bias. But then whatever we do, there will be bias. You know, I have no idea what is worse, sending it with all the problems associated, or a slightly different extraction, etc., which gives you slightly different DNA results and all that. So yeah, I'm curious to hear it in another year if you have a better handle on that part. And also getting the Europe to say you should be able to send samples freely from country to country, I doubt that will very quickly happen. So other solutions might be a bit more easy to attain. But yeah, fascinating discussion. So thank you for that part.
Yeah, thank you too. You are completely correct. It's due to, let's say, missing infrastructure of some of our partners. We in the end decided in this case to use dried soil samples, but we of course know that the drying process also changes the soil community. But at least it changes everywhere, so it's maybe again comparable. But the ideal case would, of course, be to have really fresh soil samples and then sending them around Europe. And if they are not frozen when they arrive at the lab, you again have a big problem and a bias. So I wouldn't know of any numbers to put on that. So what is the bigger bias? That labs differ in how they deal with protocols or due to shipping methods. So I can't really yet give an advice on that. But I know both things are important issues.
Yes, indeed, two big issues. Gavi, do you want to react on that?
Yes, I also think it's a really interesting discussion. I wondered a bit about the communication. So, do you have, for example, we talked about the rocks in the soil samples and also the other bias of treatments. Do you have the people, the feeling if people see a problem that you were directly contacted, so that you have been informed about it and then you found a solution for it? Or might it also be that people see a problem, think about it, and then think, okay, the best solution is this and that way, and just do it? Can you comment on this?
Yes, I think specifically speaking for the pilot, I think the communication was quite good. I think if there was any problem, I was immediately contacted and we found a solution together, or at least I got the information immediately about something that was different to the protocol, because that was something we told everyone in advance, that please, if there is something in the protocol that you cannot strictly follow, please note it in the protocol and tell us about it so that we know. But I have to say the pilot study is with 10 partners, so that's quite manageable in terms of communication. But if that would be a bigger monitoring, I don't see how you can react on every single question that might arise, because it will involve more than just 10 or 9 partners.
Yes, Ron, go ahead.
I have a question related to that communication part, because what you said, you started with the protocol, and then it turned out there were a lot of questions about that, and it took you about a year to develop, get a new protocol altogether. Of course, this is a pilot, so you have to learn from what you did. If you had to set it up now from the start, what would you do completely differently? And that's what we want to know. What did you learn that you would do completely differently if you had to set up a soil pilot now?
Well, I would definitely take more time to think about everything. I'm still quite happy about the protocol, and we didn't really change the protocol itself. We just changed the level of detail. Because we are scientists at URAC, so for me everything was clear. But many of our partners are not scientists. They are going out in the field, following our protocol, and then doing what we tell them to. And then so many questions arise that I would never think about. So I think if you really want to set up a monitoring from scratch, you need that pilot study year, at least 1 year, to go out with the protocol, test the feasibility and the applicability, and if everything is clear to everyone. I think it's not really possible, especially for such a pilot that uses physical samples also, is that you need really some time to get to know the protocol in every detail possible. So that is probably lucky us that we have that pilot study to understand all the difficulties and issues. If that protocol would have been used immediately to set up a monitoring, I think a lot would have gone wrong. Or we would have received samples that are not so well comparable because some did it a bit differently. What still is out there is that even though we have a very detailed protocol and everyone follows the protocol, we need to be aware that we need some kind of range of in which we can work and maybe adapt a little bit, like something, adapt to topography, for example, so that it has not to be the perfect square of 30 times 30. We just need to know, because from a statistical point of view, there are a lot of ways to use data that is maybe slightly off. But we need to know. So it's very important to make clear to everyone that if they don't follow the protocol in every detail, they need to tell us. So I think if I would start from scratch, I would not really do a lot differently, but I would be aware that I need that year of learning.
So you wouldn't say that, for instance, having like a workshop immediately at the beginning to get a large part of the questions would make a huge difference, because most of the questions are when they start working in the field and find out exactly, this is what I have to do now. I'm unsure what. Okay, so it's really practical.
I don't think they would have known in advance.
Okay.
Because you need to be in the field and see what really happens there. And we did have a training webinar before the field work, which was done by Soilborn. And I think it was really good for everyone to understand the main concept of the protocol, but then you need to learn in the field.
Okay, thank you.
Thank you. That was an excellent question. Really helpful to know all of this. Yeah, it highlights the importance of the coordination of the whole program and the human contact as well linked to it. You can't just have a written down protocol. Yeah, so in your experience, Julia, did this take a lot of your time coordinating the whole project?
Yes, a lot more than I thought.
Okay.
Yeah. It's because it's not also the coordination, it's also then you get data. So you need to do something with the data as well, because just because I have the samples, I know nothing yet about soil biodiversity, so it needs a lot more. And I just told you now about the fieldwork protocol, but then there is the protocol also for the eDNA or for species identification. Then you need to also analyze the data and like interpret the results and everything. So it goes on. It's not done yet after a year of field sampling.
Yeah, of course. I see there's a comment. I think it's a comment and not a question in the chat of Reidib. Maybe if you want to read it out loud or explain it a bit more.
So you need to be able to reference the soil gram content of a particular area, to bulk density. Yeah. So we will kind of measure all that, and that is what I said. Now we have samples. It's really not done yet, but such details are actually quite well described in the Lucas protocol on how then to do all those measurements of soil properties and everything. And well, soil biodiversity, I think it's such a huge topic in terms of variability of everything, and it's such a huge taxonomic range, and putting all together in one single monitoring to understand soil biodiversity as a whole and what drives soil biodiversity, that will only be a challenge, I think. But in that case, Lucas has already set quite a path. They did already also some analysis. There are some papers out there which are quite helpful. Yeah, I hope that answered the question, but it's also that in the pilot itself, probably we have not yet done everything we could and we should.
Okay, thank you so much. I see that time is almost over, so if there's a couple of last burning questions, please go ahead, anyone. We're happy to hear them. Maybe I have a question. Like, since you're on the second year of this pilot, how are you living it? Is it easier because everything is already more set up already, or do you still have a lot of issues popping up and still many things to deal with last minute? Or yeah, how is it going?
Now, I think concerning fieldwork, everything goes well now because we did have that workshop and reflected on the first year quite well. But now it's more the lab work coming up and the issues and challenges with the lab work. For example, we stick to the Lucas protocol also for the eDNA. And we got the feedback from the company that the level of detail is also much too small or little. They had really difficulties doing the same thing. And what also came out is that the primers for invertebrates are much too universal. So we only get a spotlight on parts of the taxonomy that is in the soil. So that will be the next challenge to solve on really how to use eDNA methods in such a monitoring scheme. Because on the one hand, you have the traditional methods, which are a lot time consuming and give you a great overview, but not species level. And on the other hand, you have the eDNA methods, which might give you species level on the overall range of soil organisms out there, but it doesn't work yet well. So a lot more challenges coming up.
Are you collaborating with anyone for that eDNA identification part? Because there are quite a few consortia now that do that.
Mm-hmm. On the one hand, Soilborn is still included. They know about us and we are in contact with them. Then it's the company who does the analysis. It's BioMID in Germany. They did the workshop for us and I'm in contact with them all the time. And then there's one of our partners, it's Belgium. They have their own monitoring and they are quite interested in what is happening in the pilot. And they are helping a lot with the analysis.
There's in the Netherlands, there's also the Arise project, which tries to identify all, well, like not fungi and the microbes, but all the other stuff that you can find in the Netherlands through eDNA. And they're also developing a whole pipeline how to treat that from any kind of sample that goes into identifying automatically everything that's in there. I did connect them with the ABS pilot, but they also have some expertise which might be relevant. So if you want to contact, can at least get you in touch with someone, see if they have something that you could use. Maybe.
Yeah, I'm happy to have any contacts because in the end, when we need to give a final recommendation on how to deal with soil biodiversity monitoring in Europe, we will need to decide on a lot. And any efforts that are out there already know more than I do, it would be really helpful.
Let me know. I can put you in touch with the right people over there. They try to automate everything as much as possible so that it's cheap and easy for anyone to take samples. And there are more initiatives like, I just happen to know this one, but there are more initiatives like that. So it's good to get in touch with someone.
Yes, definitely.
Yes, thank you very much for that, Ron. That is the point of these meetings as well, so we can have contacts and try to see what is being done elsewhere. So the time is nearly over now, and I just wanted to thank so much Julia for this wonderful presentation. I hope that it was useful and interesting for all of you. It certainly was for me. I got lots of insights. It was really great. And now a question for all of you who are here. If any one of you would like to give a presentation of this sort or just talk about it, please let me know. It would be very welcome to hear about your your protocols as well.
Yeah, thank you, Michelle, for giving me the opportunity to talk here. It was my pleasure.